Good morning, good afternoon, good evening. This is Robin Elliott. I'm in charge of the Parkinson's Disease Foundation here in New York. I'm here to welcome you to the fifth talk in our seventh series, what we call PD Expert Briefings, led by some of the most important and knowledgeable women and men in science around the world. I'm delighted to say that we're welcoming today for a rather demanding, at least to me, demanding but extremely important talk on gene and cell therapy. One of the experts in this area, Dr. Roger Barker from the UK, who started his career at Cambridge University, where he got his doctoral degree in neural grafting, and then moved up in the world to Oxford University, to Corpus Christi College there, where he then continued his studies, also at the St. Thomas Hospital in London. He is currently professor of clinical neuroscience and honorary consultant in neurology at the University of Cambridge, and he should be talking to us today on the subject of developing new therapies and matching them to the needs of patients in Parkinson's disease. I have, before we introduce him properly to do his talk, a couple of important business items to share with you. Many of you are old hands at this expert briefing series, so you know the drill. I'm happy to say, for a rather demanding lecture, we have 750 people already signed up for this experience. We welcome all of you, and we hope that you will take advantage both during this talk, but also in accessing it later if you want to go for a second time, as certainly I will over the next month. I'm delighted to say that we have a co-sponsorship here from, as we have for the last 2 or 3 years, with a group of independent Parkinson's organizations in the United States. The list is too long, I'm happy to say, at this point, to repeat the entire thing, but they're terrific organizations. They call themselves the Association for Independent Parkinson's Research Organizations, and they recently met in New York, actually, just last week, and so we're happy to welcome them back as sponsors for this very important talk. I also want to thank our corporate sponsors. We have 2 of them. One of them is AbbVie Incorporated, and the other is Lundbeck LLC. They've both provided very generous grants for us to stage this program, and as I hope does not need to be said, while we accept with great gratitude their generosity, they do not participate in any way in the editorial direction of the program, which is solely the work of PDF and its distinguished speakers like Dr. Barker. Just for those of you who want to download the PowerPoint slide deck, you should be able to do this from the reminder email that you should have received this morning. We obviously have your email, otherwise you wouldn't be on the call. So do download it if you want to, or you can get it from the PDF webpage, which highlights this talk, www.pdf.org. If you're one of our health professional friends on this webcast, you can earn 1 free CEU, continuing education unit, through the American Society on Aging, which has been arranged through the PDF. So if you're registered as a health professional there and have indicated you would like CEUs, you'll receive an email by the end of today with steps on how you collect the CEU. Then you've just 30 days. Please act swiftly until May 5 to collect your well-deserved unit. So now I'll turn our attention to Dr. Barker. I've already introduced him very, very generally, and I'm sure inadequately, but suffice it to say that he is a major figure in our field. We continue our tradition of inviting the best people to give talks on the most important subjects, and certainly Dr. Barker's acceptance of our invitation for this talk is no exception to this. It continues that very special, fine tradition that brings you back to our table and our webcast every month or so. So, Dr. Barker, no further introduction needed. Thank you very, very much for coming. You know the drill here. You'll be speaking for around 35, 40 minutes, and then we'll be peppering you with questions from this brainy lot who are on the webcast today, who will test you into the evening UK time. Thanks again for joining us, and the floor is yours.
Well, thank you very much for inviting me to give this webinar. What I plan to do in this talk is to take you a little bit through some of the ideas around the fact that Parkinson's disease may not be a single disorder, but possibly 2 or 3 different types of disease, which means that we need to target new experimental therapies to specific types of Parkinson's disease. So I shall mention that as by way of introduction before I walk us through really where we've got to with cell-based therapies, both in the past, in the present, and where we're going in the future with stem cells, following which I will discuss gene therapies, and I will conclude with a couple of slides telling you what we've learned so far and where it's all going in the next year or two, so that you feel more confident, I hope, about understanding how this field has developed. And I think it's important because certainly cell-based therapies has gained something of a momentum of its own, and there are many clinics around the globe offering people stem cell therapies for their Parkinson's disease, amongst other things, and one needs to be very wary of a lot of what's being offered in such clinics. So let's start with something that you will know extremely well, which is Parkinson's disease is a common disorder. That's important because ultimately if you want to attract commercialization and the development of therapies for people with diseases, common diseases attract the investment you need. So many famous people have had Parkinson's disease, and as we know, the core pathological event, it's not the only pathological event, but the core pathological event is actually the loss of a special set of nerve cells at the top of the stem of the brain called the dopamine cells of the substantia nigra. And on each side of your brain within the substantia nigra, you have around 500,000 dopamine cells. And when you've lost half of those, i.e. 250,000, you develop the motor features of Parkinson's disease, which are classically described in terms of the tremor, the slowness of movement, the stiffness, and such like. So we know that Parkinson's disease has, as part of its core pathology, the loss of a very specific set of nerve cells that release a very specific chemical, and that they are, you don't lose that many of them before you develop the features of Parkinson's disease itself. And everything that I really talk about this evening will be around trying to repair and make that system better. So it's all around trying to make the dopamine system work more efficiently, either by replacing it with cells, replacing it with genes, or trying to encourage it to grow. But as you will know, Parkinson's disease is much more than this. The pathology doesn't just sit within these dopamine cells. It extends to other areas of the brain, and it affects other aspects to do with non-motor functions, such as thinking and such like. So in order to try and better understand how to put new therapies into the clinic, the idea that you simply take it and give it to everybody, whilst it's a lovely idea, ultimately is going to produce very mixed results if we don't take into account that there are different types of Parkinson's disease. So whilst it's not a main theme of my talk tonight, this is something we set about at the beginning of the century in Cambridgeshire, a county in the east of England, where we collected everyone with Parkinson's disease over a 2-year period and have continued to follow them to the present day and work out how do people change over time. Does it lead us to believe that there may be different types of Parkinson's disease? And indeed, that is what we feel we've identified. We've identified 2 groups of patients. We've identified younger patients who may have subtle problems with thinking when they first present to their doctor, but these patients seem to have a disease which is very much centered around the dopamine system. It progresses relatively slowly, responds very well to dopamine medication, and the non-motor, non-dopamine aspects of the disease don't seem to be a prominent feature, at least in the early stages. In contrast, there are patients who have older patients, typically over the age of 72 in our study, who have some problems putting together lists of animals or categories, difficulty drawing certain objects, so they clearly have problems, subtle problems with thinking, have a particular genetic background, and these patients sadly tend to do rather less well. They have a disease which spreads quickly through the brain and involves many other systems outside of the motor system. And although I won't be talking much about these patients, this group of patients are the ideal group for many of the new therapies, which I'm sure you'll be having webinars on, around trying to actually arrest or stop the disease. So this idea of vaccines, which people have been talking about recently, a lot of the disease-modifying therapies, drugs which are designed to actually stop the disease, this group of patients are the ideal group by which to test those therapies. But what I'm talking about tonight with cells and genes are therapies which are not designed to cure Parkinson's disease. They're designed to basically deliver dopamine better to the brain. So you either do it, as I say, by replacing it with cells or genes, or you actually give therapies, namely gene therapies, which is designed to make the dopamine system, which remains, work more efficiently by giving it some form of fertilizer. So I'm going to begin by just sort of outlining roughly what these therapies are. What are these therapies that we're going to be talking about? The first is to do with cells. So as I say here, there are essentially two main types of cells that we're thinking about to repair the Parkinsonian brain. That's what I'm going to concentrate on tonight. One is the cells which are taken from the developing fetus. So these are highly controversial because you have to get these cells from abortions. You take out the developing dopamine cells from aborted fetuses and transplant them into the brains of patients with Parkinson's disease, and I'll be talking about that. The future, obviously, lies with the idea that you can do the same with stem cells. Take stem cells, turn them into dopamine cells, and then you can transplant those into the brain. People have tried other cells for other ways of fixing the brain in Parkinson's disease, which I'm not really going to be talking about. I'm essentially going to be talking about the idea of replacing the dopamine cells, and I'll come back to that in a minute. In contrast, the gene therapies are designed either to replace dopamine by essentially injecting a gene into the brain whose function is to make dopamine. So the idea here is instead of taking tablets which give you the dopamine in the form of L-DOPA or dopamine agonists, these drugs that one takes for Parkinson's disease, the idea is you inject into the brain genes which make the enzymes which synthesize dopamine, i.e. you put into the brain the genes which will allow other cells in your brain to now produce dopamine where you've injected those genes. So that's one whole area, which is to replace lost dopamine by producing dopamine more locally in your brain through the injection of genes. The alternative is to actually use factors, fertilizers for dopamine cells, which you similarly inject into the brain. But now the purpose is not for them to make new dopamine, but to make the dopamine cells and fibers that you still have in your brain work better and survive longer. So those are the different types of therapies that I'm going to be talking about. With that is also growth factors, the idea that you can actually put fertilizer directly into the brain via a little cannula. I'm not going to talk about that tonight because that's a rather separate topic. So I'm only going to talk about cell replacement therapy and genes designed to either replace dopamine or rescue the dopamine cells which are still surviving in the Parkinsonian brain. So let's start with these cell-based therapies. So that's what we're going to talk about in the first part of this talk, better ways to deliver dopamine by putting cells into the brain to replace dopamine. So this is a very simple strategy. So just to sort of recap, the rationale is that we say at the core of the problem in Parkinson's disease is the loss of these 250,000 nigral dopamine cells, these cells at the top of the stem of the brain. Now, we know that they're critical to Parkinson's disease, not only because of looking at brains, but we know when we give people dopamine drugs, they do extremely well. They do extremely well for many years. So we know that simply treating that aspect of Parkinson's disease transforms most patients for a given period of time. Therefore, a cell-based therapy, putting a cell into the brain to make dopamine, is essentially doing the same as the dopamine drugs that one takes. But the advantage here is that the dopamine is being replaced specifically where it's needed. So you're replacing it at the site where it is lost in Parkinson's disease, and you're replacing it with a nerve cell which will release dopamine in the normal way that a dopamine cell releases dopamine. Now, that's important because the side effects, which often limit the usefulness of the dopamine drugs, are due to problems with them acting on dopamine systems outside the area of greatest loss, so not within the what they call the striatum, but affecting other areas in the brain, which can make people behave indifferently. So these behavioral problems you see with the dopamine agonists, it can cause people to start seeing things and getting confused. And all of that is related to the fact that the dopamine agents are working at sites outside where they need to work and causing problems. Cell therapies, by putting the dopamine cells just where you need them, you will avoid those off-target problems which you see with the oral drugs. Secondly, the big problem with the dopamine drugs is after a period of time, you get these motor fluctuations where the tablets work or don't work. When they do work, they create these involuntary movements, these dyskinesias. And that almost certainly relates to the fact that when you're taking the oral dopamine drugs, they stimulate the brain very effectively, but they don't stimulate it in the normal way. And that so-called non-physiological stimulation of the brain with the oral drugs sets in motion a series of changes within the brain, which leads to the development of these involuntary movements, these dyskinesias. Putting dopamine cells back in the brain in the form of new nerve cells will ensure that that dopamine is released at the site where it's needed, and it's released in a way which is the same as the normal dopamine cells, i.e. physiologically. So in theory, it should not only just work where it needs to work, but it will avoid the long-term complications which you see with the oral medication. So the hope with these cell-based therapies is not that you will be cured with Parkinson's disease, but it will dramatically change the natural history of treated Parkinson's. So if ultimately we can do this early on in the disease, it will avoid patients taking any drugs, it will avoid the long-term complications with the drugs, it will avoid the need for the more advanced therapies which are out there, such as deep brain stimulation, and it will therefore be very transformative for many years for many patients. So that's the rationale. That's the reason why we're interested in cell-based therapies, to try and help people with Parkinson's disease. Now, obviously, when you're trying to develop something from an idea to take it to the clinic, you have to work out how to do that in the lab and then translate it to the clinic. And I'm not going to spend a lot of time talking about this, but it is important to understand it because a lot of therapies which are currently being touted as useful for Parkinson's disease, which are cell-based, have not gone through this rather simple procedure. So the essential way in which you take a cell therapy to the clinic is you identify that cell which you wish to use. In our case, we're looking at cells that make dopamine. We test them in an animal model of Parkinson's disease, which creates problems in its own right because the only animal on the planet that gets Parkinson's disease is man. So if you want to test it in the lab, that's a problem. But actually what you really want to test for the therapies we're talking about is an animal model that loses dopamine, and there are very good models for that. If you can get success using these animal models of dopamine loss in the brain with your cell-based therapy, that enables you to then say, let's try it in a few patients in a so-called open-label study. And an open-label study is essentially a study where you know what you're getting, the doctor knows what you're getting, everyone knows what you're getting. It's open. Everybody understands that the therapy has been given. And that obviously creates a problem of bias. The investigator wants you to get better. The patient wants to get better. Everyone wants to get better. And so that can create so-called placebo and investigator bias effects. I have to say I think that is slightly overplayed in a lot of trials because the condition, as you know, is a progressive one. So having long-term sustained benefits is very unlikely to be due to any placebo effect. Nevertheless, that's how you do it. You do it in a few patients, see what result you get, try and work out whether you've got any response. If you have, can you get it better? Then you optimize what you're trying to do, and eventually you go to these so-called double-blind sham surgery trials, which are the sort of gold standard that people use, where essentially you have patients who have surgery, some have transplant, some do not. The patient doesn't know whether they've had a transplant or not. The doctor assessing the patient doesn't know whether they've had a transplant or not. The only person who knows is the surgeon who did the surgery. And therefore, everyone is blinded to what's going on, so you will see a real effect. And that's the process by which you need to develop a therapy from an idea to actually something that could become a mainline therapy in Parkinson's disease. Now, one of the problems in this field is that people rather rush through aspects of this, so you can end up coming to premature conclusions or premature trials and arriving at results which might not be entirely correct. So this approach of taking a cell source to make dopamine for Parkinson's disease as a way of treating Parkinson's disease has been tried with many different cells. But the only one which has really stood the test of time are those which have been taken from the developing fetal brain. So this is obviously easy to do in the lab. In the clinic, you need, as I say, to get it from aborted fetuses, typically age 6 to 8 weeks. But essentially what you do is you then dissect out where the normal dopamine cells develop, which are the ones lost in Parkinson's disease. And in animal models, if you dissect that out, so in the rat, if you take out the developing midbrain where these dopamine cells develop, you then transplant them into an animal model of Parkinson's disease with a loss of dopamine on one side, which is this so-called 6-hydroxydopamine. This is a very standard model. You knock out dopamine on one side of the brain, and then you transplant in the dopamine cells which you've prepared from the fetal midbrain into the area of the brain where dopamine works. And experimentally, and this was done in the 1980s, it was demonstrated if you did this in rats in many labs by many different people, you took the dopamine cells from the developing fetus, transplanted them into the brain of these animals that had lost dopamine on one side, the cells survived indefinitely. In the case of a rat, that's about 6 to 9 months. That these cells not only just sat there, but they connected and made connections with the brain and received connections from the brain, so they actually wired up into the brain. They released what you wanted them to release, which was dopamine, and many of the behaviors that the animals had when you knocked out dopamine on one side of the brain was much better. And in this picture here, all of the brown you see in that picture is in fact transplanted dopamine cells. The brown is the dopamine cells and the fibers that grow out of it. So we knew by the mid-1980s that if you took tissue from the same species, which is what allografted means, so rat to rat, human to human in the clinical situation, you took it at the time at which the dopamine cells normally developed, which in the human situation is around 6 to 8 weeks, and you placed it where dopamine works in the striatum, you could get fantastic effects in the animal models. So in the mid-1980s, this then moved, as I'd already said, into open-label studies. So this was first done in Sweden in the mid-1980s, and the first 2 patients who were transplanted showed no benefit whatsoever from this approach. So you could conclude that this was a great idea but didn't work. What they did was they said, I wonder if it didn't work because we didn't put in enough tissue, the way in which we injected it was wrong, so they modified their technique, and that's something we're learning all the time, how to learn as you go along to modify what you're doing. And then in 1988-89, 2 more patients were transplanted. And then with these 2 patients, the whole field suddenly came alive. So in these 2 patients, patients number 3 and 4 who were transplanted in Sweden, one saw a dramatic response, especially in patient number 4. So number 4, although it's hard to see on this graph, before they had their transplant on one side of the brain, spent 2/3 of the day off. In other words, they spent 8 out of 12 hours as though the tablets weren't working and they were unable to move and really live a normal life. They then had a transplant on one side of their brain, which was derived from these developing dopamine cells from the midbrain from 3 to 4 fetuses. And over the course of the first few months, nothing much happened. However, over the course of 3 years, the patient went from being off 2/3 of the day to being on all day. So having not been able to move for 8 out of 12 hours, they could now move for 12 out of 12 hours. That was a dramatic clinical response. It's even more dramatic when you realize that over that 3-year period, all of their medication was actually stopped for their Parkinson's disease, apart from selegiline, which was flavor of the month in the early 1990s. Although I won't be talking about it, that patient actually died a couple of years ago in Sweden in his late 70s, and at post-mortem he has a perfect surviving transplant in the area where those cells were put in 1989. So 24 years after he had the transplant, the dopamine cells are still very happy surviving and innervating the brain. So this patient, anyway, in the early 1990s showed that you could get a significant clinical improvement. So the patient was clinically better. Just like the animals got better with the transplant, now the patient got better. They then took that patient to London and they scanned the patient's brain to look for dopamine within the brain. So you can do scans called PET scans where you can look at the chemical in the brain. And what they found was not only had the patient improved clinically, but on imaging at the site of transplant, the dopamine signal slowly increased back to normal over that same 3-year period. So that showed the clinical improvement matched to the dopamine levels in the brain, which gave you very strong evidence and confidence that you could actually repair the Parkinson's brain and actually make patients dramatically better. Now, this was adopted in some places in America in the late 80s, early 90s, one of their patients who showed a more modest response but died 18 months after a transplant through a heart attack, when they actually died and they looked in their brain, they could show not only had there been a clinical improvement and imaging improvement, but actually when you looked at the anatomy, histology of the brain, you could see surviving dopamine cells at the site of transplantation. So you could now say that this approach in a few patients worked in some cases. That clinical improvement was matched by evidence of dopamine surviving cells in the brain. And furthermore, in a study that was done a few years later, it was demonstrated again by the team in Sweden and in London that those dopamine cells not only survived, they not only produced a clinical response, but when you actually stimulated the transplant to release dopamine, they released dopamine in exactly the same way as the normal dopamine cells. So this demonstrated that you could get cells to survive, you could get them to release dopamine normally, and patients got better. Now, the problem really came about in the early 1990s with this whole field when George Bush Sr. left the White House, Bill Clinton came into the White House, and federal funding was released for this type of work in America, the consequence of which was that the National Institute of Health in America funded 2 trials using this so-called double-blind placebo-controlled approach. This was done, I should say, in the early 1990s at a point when we had yet to optimize and fully understand how this therapy worked. So many people saw these trials as being premature because we didn't know how to do it properly. And if you jump straight to these type of trials, the results of it are clearly going to be held in much higher regard than those from these open-label studies because of the nature of the trial design. And that indeed is exactly what happened. So these papers, which then published their results in 2001 and 2003, both reported similar findings, which was that if you transplanted dopamine cells into the brains of people with Parkinson's disease and compared it against people who did not have such a transplant, they did not work. Now, they didn't work in the trials that they designed. The first trial, that endpoint was they phoned the patient up after a year and asked if they were better. In the second trial, they used a small amount of tissue, a large amount of tissue, and waited 2 years to see whether there was any benefit. And although there was a clear trend towards a benefit in those who had the bigger transplants, overall there was no significant effect. Furthermore, not only was there no significant effect, but some of the patients now started to develop some of these involuntary movements, which you see with everyone with L-DOPA, but now you were seeing them in the context of a transplant. Of course, if you get them with the L-DOPA drugs, you can get rid of them because you can get rid of L-DOPA. If you get them with a transplant, you have a problem because you can't retrieve the transplant. They were amenable to treatment with deep brain stimulation, but nevertheless, there was now a side effect with this therapy which hadn't been reported before, and that was in the context of a trial, 2 trials, which showed that it didn't work. So that raises a question, what's going on? I've told you that in these small open-label studies in some places in the US and in Europe, you got dramatic responses with long-lasting benefits. Some of the patients in the UK who were transplanted in Sweden are now 25 years after their transplants, 30-odd years after they developed Parkinson's disease, and have a motor score that is less than when they presented over 30 years ago on no treatment. So you have some open-label studies that have shown dramatic responses, and you have these double-blind placebo-controlled trials that tell you it doesn't work and it creates side effects. So who's right? Who has told us what actually goes on with cell-based therapy? Well, the problem is that neither of them have told us what the answer is because both or all of these studies have actually looked at things in such different ways, which tells you something about how the field has developed. If we all knew how to do this, every trial would essentially look the same. But actually, if you start to compare these studies, so here we have the double-blind studies, the American studies funded by the National Institute of Health, and this is the biggest open-label study done in Sweden. So how did these trials compare? Well, first of all, they used different types of patients. Some used young patients, some used old patients, some used early-stage patients, some used later-stage patients. So none of these trials used the same type or subtype of patient. These trials all used different doses of cells. Some used a small amount, some used a large amount, some used even larger amounts. So they all had different doses of cells. One of the studies delivered the cells by going through the front of the brain. The others went from the top of the brain. So they were delivered differently. Some of the trials used drugs to suppress the immune system from rejecting the cells. Some did not. Some used them for 6 months. Some used them for 2 years. So the levels of immunosuppression were very different between them. They used different endpoints. As I say, one of the NIH studies used 1 year, but I've already shown you data to say that the patients in the open-label study took over 3 years before they noticed their maximum benefit. So they used different endpoints at different times, and they used different follow-ups. So just to sort of try and make this point about why this is so important is if in the early 1950s I had taken 40 people with a chest infection, just any old chest infection, and I'd given them 300 milligrams of penicillin and done a chest x-ray after 5 days, what would I have found? Well, what I would have found was that the penicillin did not help the chest x-ray evidence of a chest infection. And the reason why that would have failed is because I used the wrong antibiotic at the wrong dose in a group of people who had a whole mass of different chest infections with a chest x-ray which might not be the right endpoint by whether to see whether the infection's gone away. Now, if I just believe that one trial, having given people 300 milligrams of penicillin versus nothing with a chest x-ray at 5 days in 40 people with a chest infection, I could have come to the conclusion that penicillin doesn't help chest infections, therefore penicillin doesn't help microbes, therefore antibiotics don't work, let's abandon them. Now, I know it's an extreme example, but it makes the point that when you see trials that don't work, it doesn't mean that the therapy doesn't work, it just means the trial hasn't produced a positive result, which may be because the drug doesn't work or the agent or the cell doesn't work, but it may be because there's something a bit more fundamentally wrong about how you've done the trial and delivered the therapy. So in a way to try and take this forward, we set up in Europe and in America, we invited everyone together to go back and look at all the data we'd collected over the years in all of these transplant trials to try and work out why it was that some patients had improved and some had not. So we went back and put all the data together. We have a plot here of all of the 2-year data on all of the patients who were transplanted in the centers that would provide us with the data. And what we could show was that there were clearly a group of patients who had a sustained and marked response to the transplant, about a third of the patients. And those patients that seemed to benefit most from this whole approach were those who were younger, so under the age of 35, with less advanced disease. So the earlier you were in the course of your condition and the younger you were, the more likely it was that these therapies would respond, that you would respond to these cell-based therapies. In addition, because we had these problems now with these graft-induced dyskinesias, what was the cause of that, and could we mitigate against that if we were going to go forward? Well, 2 theories emerged as to what caused these involuntary movements driven by the transplant, so-called graft-induced dyskinesias. One related to the fact that the dopamine cells in some trials were not grafted in a way that meant they were evenly distributed across the area they were supposed to. So you had these little hotspots of dopamine cells, which meant that bits of the brain were getting too much dopamine and others were not, which may be driving some of these involuntary graft-induced dyskinesias. The alternative, or theory which also was put forward, was that actually the transplants contain another group of cells, not just the dopamine cells, but cells which produce a chemical called serotonin, or 5-HT, which is involved with mood and sleep and such like. But these cells can release dopamine in a way which isn't very regulated. So they can make dopamine and release dopamine, but they can't take dopamine up. So the theory was that the graft-induced dyskinesias, which were a major side effect related to how the cells were delivered, and actually that the cells were the transplants were contaminated by large numbers of these so-called serotonin or 5-HT neurons. So if you then take that information gleaned from just simply reviewing what's gone on in the previous 20 years, you can then start to put that information together and build a new trial, which is what we did, and we're fortunate enough to get funded through the European Union, and this is an ongoing trial that I coordinate at the moment. So what we did was we selected the patients that I've told you about, so those who are younger, less advanced, without so many of the L-DOPA-induced dyskinesias. We enriched our population for that. We collected 150 of these patients, which we followed to the present day, doing 6 monthly assessments with them wearing baseball caps so we can video them and send the videos to others to rate, who are blinded, therefore, to whether the patients had surgery or not. So we've standardized the patients. We've standardized how we collect the tissue so that we can increase the number of dopamine cells and minimize the number of the contaminating serotonin neurons. We've developed a transplant injection technique, which was developed in Sweden, which means that the cells are evenly distributed across the area where we want them, so we avoid these hot spots. And rather than do sham surgery, what we've done is we've taken these 150 patients, we've randomly selected 20 of them to have a transplant, we've randomly selected another 20 to have some expensive imaging to match with that, and those grafted patients will receive the transplant, and because they're wearing baseball caps, we can send, as I say, the videos to other people who will then be able to assess patients, not knowing whether that patient has had a transplant or not, and even if they have, whether it's before or after that. So we feel that gets around a lot of the bias and the need for sham surgery. So we've designed the trial so that we can recruit patients in that way. We're going to look at these patients at 3 years, which is the time at which we think we will get the maximum benefit from these transplants, and we will look at lots of different measures by which to assess whether they're useful or not. We are currently at this stage where we are now transplanting patients, and since May of last year, we have actually transplanted 5 patients with 10 transplants, 1 to each side of the brain in 5 patients, and we hope to finish that trial by the end of this year. Now, this trial has taken, obviously, a long time to get here. There are problems with using fetal tissue outside of the ethical problems to do with tissue availability. You need 3 to 4 fetuses to do one side of the brain. And so as a result of that, we have always realized that this is a stepping stone, if you like, to the future, which is with stem cell-based therapies. So what are the stem cell-based therapies we're going to do, and why do we consider them? Well, I've already mentioned some of these. There's obviously logistical advantages. If you have stem cells, you can grow them in the lab and have an endless supply. They're easier to control, so you know at the end of the day you'll be injecting actually a population of stem cells that you hope have turned into dopamine cells, and they are slightly ethically less contentious, but they're not without ethical problems. So that's why stem cells have been attractive. The ethics around them is less contentious. You can produce more of them. You can control what you're doing with them. And when you ultimately inject them in the brain, you know exactly what you're injecting. Now, the question is, which stem cells are we thinking of? Because there are lots of different stem cells that are out there. And although there are many different types of stem cells, and the ones which you'll see widely advertised in clinics are mesenchymal stem cells and other adult stem cells, I would say that there are essentially only 2 main sources of stem cells that are likely to come to clinic in the next few years. One are the so-called induced pluripotent stem cells. So these are stem cells where you take an adult cell, such as a skin cell, you can take that skin cell, you can reprogram it back to its origins, i.e. back to a stem cell, and then you can take that stem cell and you can drive it to a dopamine cell. So people have been very attracted by this because obviously you could take patients' own skin, turn it into induced pluripotent stem cells, turn them into dopamine cells and transplant them back into the patient's brain. Now this has been done in rats, so it's been shown to work in rats. It's been shown to work in non-human primates in Japan. And now there's also this exciting idea that you don't need to take skin cells and turn them back to stem cells to turn them into dopamine cells. You can directly turn them from skin cells into dopamine cells, so-called induced neurons. And that's also been shown to work in animal models of Parkinson's disease. Now, this is very attractive, and it's very attractive particularly to people in Japan where they're pursuing a lot of this. I think the question you have to ask yourself is, first of all, the cost of making individual stem cells for a person is prohibitively expensive. And secondly, would you want to put back in cells which you've reprogrammed to make dopamine cells from cells which contain all the genetic information, whatever that is, to give you Parkinson's disease in the first place? So there are worries about actually whether you'd be putting back cells which ultimately are not the best cells. And more recently, there have been anxieties around the stability of these cells. When you reprogram them, do you introduce mutations which may make the cells somewhat unstable? So whilst this is actively being pursued, I would say that the front runner is much more likely to be these embryonic stem cells, these cells which you get from spare embryos and which all of us began life as embryonic stem cells. And the reason why these are attractive is that 5 years ago, a group in New York managed to demonstrate that if you take human embryonic stem cells, you could actually turn them into dopamine cells, which look like the dopamine cells which you need to replace those lost in Parkinson's disease. And my colleagues in Sweden, Malin Palmer, managed to do the same with a refined protocol. Not only could they turn the embryonic stem cells into dopamine cells, but those dopamine cells, when you transplanted them into the brain, made fantastic connections, released dopamine, and in fact behaved just like the fetal dopamine cells, which we knew worked in patients already. So this has led us now to believe that we're actually on the verge of going to clinic with these cells. Now, learning from the past, we obviously want to do this in a much more considered way and try and avoid the mistakes of the past. So as an initiative to try and do this, we've set up a group which collects all the major groups around the world who work in this, in Japan, in Europe, and in America. We have meetings on an annual basis to try and discuss all the data so that we make sure we can patrol and protect each other from doing premature trials and understand and learn from each other. And the consequence of that is that we imagine that within Europe we will be going to trials in 2 or 3 years' time, in Japan next year or the year after, and in America it's felt by the New York group that they will be doing it around the same time as in Europe. Not only does this group allow us to learn from the past, but it also ensures that we can at least make comments on other trials which are taking place. So this is a trial which is planned in Melbourne in Australia, run by a company called ISCO. They have developed a particular type of stem cell which has not really been adopted by many groups for various reasons. They are planning to transplant 12 patients with Parkinson's disease. They have got permission to do this, and so they are at the verge of starting this trial, even though the preclinical data, which I told you about at the beginning, actually is not there to support their use. So as part of this, we've actually written an article which appeared in the Journal of Parkinson's, which goes through some of the arguments I think one needs to take into account when considering whether these are the type of therapies one should be using in the clinic. And the reason why we write such commentaries is if this trial goes wrong, then the whole field will essentially be derailed and come to an end, even though we think that this is not the optimal cell therapy to take forward for the reasons that I've said. Now, I'm rather running out of time, so I'm going to quickly go through gene therapy just to make the same points, because what's happened in the field of gene therapy for Parkinson's disease is very similar to what's happened in the field of cell replacement therapies. So in this particular approach, I'm going to talk briefly about genes which have been designed to replace dopamine. So this is gene-based therapies. So in the case of prosavin, which is a gene-based therapy made by a company called Oxford Biomedica, we were involved with this trial. Essentially, this injects genes into both sides of the brain that makes dopamine. In this trial, which was published 2 years ago, it was demonstrated that the gene was well tolerated, it produced no major problems, the surgery was safe, and that some of the patients showed a significant improved response, the consequence of which is that Oxford Biomedica are in the final stages of planning a trial which will begin in Europe at some point in the next few months. Now, as an alternative strategy, which I mentioned at the beginning, is rather than replacing dopamine either in the form of cells or in the form of a gene, the alternative is to give some sort of fertilizer to make dopamine work more effectively. And the best factor for this is a thing called glial cell line-derived neurotrophic factor, GDNF, which was subject to a growth factor trial where it was injected into the brain, and it produced mixed results. Now, that factor was owned by a company, so another company decided to try gene therapy, but weren't allowed to use GDNF because it was owned by the company. So went for son of GDNF, which is an agent called nurturin. They did their open-label studies. They went to their double-blind, placebo-controlled studies where some people got nurturin, some just got saline injections into the brain. And what they found in this study was that there was no major effect at 12 months, but there was some indication that there may be something happening a little later on. Not only did the patients not improve on their clinical measures, there was nothing seen on the scanning which worked so well to demonstrate benefits with the cell-based therapies. Undaunted by this, they argued that one of the reasons here was perhaps they hadn't quite targeted the system properly, perhaps the agent wasn't getting into the brain properly, so they planned another trial where they went for slightly earlier stage patients, injected it into 2 sites in the brain, and published their results last year. So this is the paper that appeared last year, and what they found was it didn't work. So here was a second study using a gene therapy with a growth factor which failed to work. So why didn't it work? Is it because the gene therapy itself is a failed therapy? This whole approach of rescuing the dopamine brain by giving growth factors is a waste of time. Well, that may be the answer, but I think there are other explanations. The first thing is that they may have been choosing people at the wrong disease stage. Obviously, if you want to give a growth factor, then you need it to work on a system where there's something to grow. So generally speaking, these growth factors are given to people who've got relatively advanced disease. You would be much better off giving it earlier on, because earlier on is when you have dopamine fibers to rescue. So basically, if you pour a fertilizer into a field which has no seed, nothing will grow. There's nothing wrong with your fertilizer, there's just no seed to grow. The same applies here. If you put your growth factor into a brain that has no fibers to rescue, you will rescue nothing. And indeed, when they looked at some of their earlier stage patients in this trial, there appeared to be a response if they'd gone earlier. So one of the reasons this trial may have failed is because they used the wrong type of patient. They should have gone for patients with newly diagnosed Parkinson's disease. The second problem may have been that they gave the wrong dose. And when they published the paper last year, you can see this in that on this picture here, the distribution of the growth factor in the brain was extremely limited. So it only ever went into a very small part of the brain and therefore only had limited potential to rescue the system. And finally, it's recently been discovered that the receptor which binds that growth factor, so the growth factor hits the receptor then mediates the benefit, that receptor itself can be affected by the disease process itself in Parkinson's, which means that you may need to think about how you can improve that by giving drugs with your growth factor to improve the system. So to conclude, what have we found up to 2016? So what we have found is that cells can replace the lost dopamine cells. We've shown that with fetal dopamine cells. They can produce dramatic benefits, but it's inconsistent. Can we do better? We probably can, but ultimately this is a therapy that's never going to become mainline treatment for the reasons I've said. Stem cells are very promising for the future, especially now that we can make differentiated dopamine cells of the type needed to replace those lost in Parkinson's disease. What about gene-based therapies? Well, we've shown that gene-based therapies can actually improve in the short term when you replace dopamine. We've shown that when you give growth factors, they don't work, but there may be other reasons for that, and new trials ought to be done, even though the company that made GDNF, made Nurturin, has now given up on this whole approach. So what does the future hold? What lies beyond this year and this year? Well, as I've told you, we're undertaking a fetal transplant trial at the moment. We hope to finish transplanting at the end of this year. The 3-year endpoint will take us to 2019. We will tell you the results in 2020. Stem cell trials are being planned across the globe in the next 2 or 3 years. I think they need to be done in centres which understand what they're doing rather than many of the other places which offer this. And I won't mention the trial they're doing in New Zealand tonight. The gene-based therapies, Prosavin is now in the final stages of going to clinical trial this year. There's a new gene therapy being developed by the team in Sweden that they hope to take to a clinical trial next year or the year after. There is a GDNF gene therapy trial which is currently underway in America, which hopefully will publish its results soon. So to conclude, what have we learned? I think if you understand that Parkinson's has different types, we will understand how to better target our therapies to actually patients with different types of disease. These therapies are not for everyone, they're only for some types of patients. I think we need to really rethink about how we use these therapies, and actually a lot of these agents might actually be better used, not in the more advanced cases, but in earlier stage disease. And that maybe ultimately prove to make these therapies much more competitive and effective. And finally, I would say that all of these therapies will be coming soon to clinic. Ultimately, though, they'll have to compete with what's out there. And I do believe that if we can actually understand how to better trial these therapies in the right group of patients at the right stage of disease, learning as we go along, they will ultimately become first-line therapies for Parkinson's and will be competitive against everything else that's out there. And finally, I'm just going to thank all the people who've helped me with some of the work that I've talked about over the last 40 minutes, and thank you for listening.
Dr. Barker, thank you very much. That was just terrific. My goodness, amazing. And those of you who were looking at the slides, as I was, certainly want to spend a few hours reviewing them and doing this over following the session. Very remarkable presentation. I should tell you, I'm not sure this is a compliment to you or to our participants here, that actually almost more than half the questions we received, you answered during the course of the talk, which either shows that they were terrific questions, or it shows that you are very perspicacious in the way that you organize your answer. I suspect it's a combination of both. But I've still managed to retrieve a few that you did not answer directly, and so I will lead on those. We have a few minutes to go with this, and for those of you who still have a question pressing and would like to ask it now, if we don't get time, we certainly will endeavor to answer it later. Please click the text box on your internet connection, enter your question, and then click the send button. And then we'll answer the question as the time permits. So do ask along and ask ahead, and we will endeavor to, if not answer it now, at least later on. Here's a question from New York. I hope I'm getting this right, but even if I'm not, I think it's an interesting question. It has to do with this amazing array of experiments that have been going on now for 25 years. Given that in people with Parkinson's, there's something going on that creates the condition they have, and the particular question from New York is saying we're now obviously understanding that alpha-synuclein may be at the core of this kind of problem for the people, for many of the people who get Parkinson's. Then what is that going to mean, even if you were to perfect a surgical technique with replacement therapy or gene therapy, wouldn't you still have in the potentially affected brain precisely the kinds of things going on that would kill what you're doing?
Yeah, so this is a very good question, and in fact I didn't mention it, but it's been recently discovered in the last few years that in some of the patients, especially when they've had the transplants for more than 10 years, that the transplant itself acquires some of the pathology of Parkinson's disease. So some of the grafted dopamine cells develop alpha-synuclein pathology, as you'd see in the Parkinsonian brain, and that sparked a whole new theory on what may be underlying Parkinson's disease. I think in answer to the specific question, though, that rate of accrual, if you like, of the Parkinsonian pathology in the transplant is very slow. It's about 1% of dopamine cells per year. So ultimately, the transplant will probably succumb to the same disease process as the Parkinsonian brain, but in order to lose sufficient number of cells for that to happen, it would probably take 50 or 60 years, by which time something else would have overtaken the patient. So I think it's important to understand that these transplants will acquire the pathology at a low rate. It will ultimately affect their efficacy, but not for many decades. And that the therapy I'm talking about is not exclusive. So if someone comes up with a treatment which will stop Parkinson's disease, then there's no reason to think you couldn't take a patient newly diagnosed, put in some new cells, and give them the therapy which stops the disease, and they can be then complementary, these two approaches.
Great. Thank you very much. This is a question again from New York. We have an unusual number of people talking from New York today. They're very garrulous around where we are, but they're very good questions. This person actually is asking a sort of more of a medical economics question, and I realize that despite the fact your skills are amazing, and medical economics is not necessarily one of the things in your resume, but this person is asking a question that I'm going to expand on a bit myself that makes it more connected with your talk. Question is, the therapy is clearly going to be very expensive, and as you pointed out, the resources, both in gene therapy and cell replacement, are by their nature limited. So the cost per patient is likely to be very high. This person from New York would be interested in your view of that, what that means. But if I can extend the question a bit further, is it possible that the work going on in your work and others who work with you is going to be more important in therapies in the future for what it teaches us about a new generation of therapies that may build on the science that you're creating rather than the application of the cell replacement and the gene therapy techniques themselves, which inherently are always going to be very expensive. Do I make that clear?
Yeah, no, I think you're absolutely right. So I think we learn a lot through this. So we will learn a lot about what makes a dopamine cell from a stem cell. And by the same inference, you can say, well, what makes it die in Parkinson's disease? So learning the techniques and the technology which allows you to make the cells may also instruct you about what goes wrong in Parkinson's disease. Now, I personally don't think that cell-based therapies are necessarily expensive. So, for example, with the protocol developed by the team in Sweden, you can take a 6-well plate, so a normal laboratory dish with 6 small wells in it with the embryonic stem cells in it, and from that 6-well plate over 3 weeks, you can make enough dopamine cells to transplant 750 patients. So you don't actually need, so if you had 100 plates, you would be able to do whatever it is, 75,000, which would be a significant number of patients. And that production cost of that would actually be relatively low. Now, it does raise a very interesting question, which is that however successful I am and my team and the people I work with at taking this to the clinic, ultimately this therapy to become a mainline therapy will need to be bought up by a company. And then the question is, if it's very effective, then it could be very expensive, not because it costs a lot to make, but because you only need one injection at the beginning of the disease and you need nothing else. And therefore, in order to recoup their money, they're going to have to charge you a lot up front. But I don't believe that the cost of making these therapies is ultimately going to be very expensive. I think the commercialization and the payback to the companies that invest in it is likely to push the price up more than the actual production itself.
Great answer. Thank you very much. This person, also from New York, I'm afraid to say, is asking a question about the dyskinesias that you referred to in the early part of your talk, and were one of the serious side effects of the earlier experiments that were done with cell replacement. And this is a bit off the topic, but relevant enough, I think. She would like to know, she's heard apparently that the incidence of dyskinesias that have complicated and made less attractive some of the results of the earlier trials may have themselves stimulated new research on therapies for dyskinesias, therapies that will stop or reduce dyskinesias. She wants to know, is that true? Are we getting anywhere in controlling dyskinesias, whether or not they're directly related to the surgical therapies we're talking about now?
Yeah, that's absolutely spot on. So one of the, which I briefly mentioned, one of the explanations as to why people may be developing dyskinesias with transplants relates to the fact that these so-called serotonin 5-HT neurons mishandle dopamine, release it, and that's what drives the dyskinesias. If that is true, then if you give drugs that silence the serotonin, the 5-HT nerve cells, then you should be able to deal with the dyskinesias. And so there has been a whole new therapeutic avenue opened up targeting that particular type of receptor. So graft-induced dyskinesias has reopened the debate as to what drives levodopa-induced dyskinesias, which in itself has led to new therapies around a different mechanism.
Thank you very much. I think we're time maybe for a couple more questions. There is somebody, oh yeah, here's a good one. Somebody from Illinois is asking the question, we have many people here, Dr. Parker, it won't surprise you, on this call who themselves have been involved in research themselves by volunteering for trials. In fact, I found somebody we were writing a little citation for in our own organization over the weekend, who is somebody who's been very active among our patient engagement experts, who's been involved in 25 trials over her life. So they're very serious contributors, as well as people who obviously look eagerly at the possibilities of help. So this person from Illinois would like to know, for the average Joe or the average Jane sitting here in the States or in the UK or anyone else listening to this call, what opportunities are there going to be for participating in research of the kind you're talking about? I think you mentioned that you have 150 people, subjects in your own trials, if I got that number right, which is much larger than anything I've heard in the earlier trials that were going on. Does it mean there'll be more opportunity for people who like to participate in research to present themselves as candidates for your work?
Yeah, so I think one of the big problems which has happened in this field is that I think there hasn't been enough follow-up on the patients. So one of the sadnesses for me is that many of the patients who were transplanted in America, for example, in their studies were never followed up long-term. So we don't know what happened to all the patients who were grafted in the 1990s because no one's been collecting them and following them up. My own view is that for the average person with Parkinson's, the best thing you can do is get yourself into a research clinic. And I'm a great believer, this is what we're trying to do here, in setting up large cohorts of people, following them over time. And then as new therapies come along, we offer them to the patients we think are most optimal for that. And then we can compare our therapy against best medical treatment, surgical treatment, or other experimental treatments. So our plan is, for example, with our stem cell-based therapies, when we go to clinic in 2 or 3 years' time, we will probably take some of the patients who haven't had the fetal transplants but have been followed up as part of the observational arm of that study and transplant them. And then we can ultimately say, how do the fetal cells do against the stem cells against other therapy? It is difficult for people to get into trials, and I sympathize with that. I get a lot of requests here to come and join the trials. People want to buy themselves into trials and such like, which obviously is not allowed. I think making sure you're at a research active center, taking part and listening to webinars such as this, greatly empowers you to know what's going on, and then you can search out whether it's possible to be involved in research at whichever site offers that particular therapy.
Terrific. Last question is also from Illinois, and it's a question I would love to hear you answer, even though it's a little unfair because it deals with this whole scope of economics and medicine together. But the issue that this person from Illinois would like to know about is whether we're putting enough resources in general into this field, which by your account today has certainly for me has made me feel even more confident and excited about this particular avenue of research. I do remember the time in the 90s and the early aughts where there was great disappointment in this, and things, there were obvious failures, as there always are in research, but it seemed like there was even a possibility that there would be blind avenues and blind alleys here. And obviously you're telling us that particularly in the European initiatives and the way that you have this it's almost this multinational disease-oriented consortium almost, if that's not the wrong word, to look at this field as a whole. Clearly things are really much more exciting than perhaps they were just 10 years ago. Are we, in that context, spending enough through the NIH, through the European research agencies, through the Japanese agencies, the three principal engines of research funding in the world, doing enough to encourage this? Are enough scientists involved? You mentioned a cadre or a coterie of a couple of dozen people working around your field. Should we be in our role as policy advocates, which many people on this call are today, including the people at the Parkinson's Disease Foundation? Should we be urging more attention to this? What's your feeling generally about resources to meet the challenge? We understand that you are potentially compromised in the best possible sense of that word because of your own very successful work. But I can't resist the opportunity to finish the talk by asking you what you think of resource allocation and this area of research.
Yeah, I think it's a very good question, and it's a very difficult balance. It's resources and regulation. So some people say, is the regulation too much? I think certainly in the UK, of course, stem cells have been very much at the front of government thinking for many years. So the UK is a very fertile ground, if you like, for funding this type of research, as is Europe. And America, I would say, has been slightly less inclined by virtue of some of the trials that have taken place in the past. I think that there is funding going for this. I think going forward, I think people are looking at it. I think it's important to do it in a measured way. So I'm a great believer that teamwork and collaborative work is necessary to make this happen. It's not about the first person past the post. It's not the first person to go to a trial. It's about getting it right and learning as we go. And the most important thing, I think, is understanding that when you do this for the first time, you're likely not to get it right. And people forget that. So when we're moving forward, it's very important, I think, for everyone to understand we're learning as we go. When Christian Barnard did the first heart transplant, that patient lived for a week or two. It made materially no difference to their life, but that triggered a whole series of experiments in people and work, which ultimately led to whole organ transplants as we know it today. So I'm a great believer that this therapy will go forward, that the funding for it is looking very good at the moment. You have to make sure that you're funding the right programs with the right combination of clinicians and scientists. I think the patients have a vastly important role in all of this in terms of understanding what it is we're trying to do. And I was actually down in London talking to the Cure Parkinson's Trust yesterday, and I was saying, coming back to one of the questions earlier on about the cost of these therapies, ultimately, if these therapies do come in and they're very expensive, you as the patient are in the strongest possible position to argue for these therapies to be reduced in price and to promote their adoption. Because as scientists and clinicians, we can shout, but people generally don't take too much notice of us, but the patient community actually is terribly powerful at what it can do here. And so I'm very grateful for the opportunity to share our work with such a community, knowing that that community itself can help shape the future of how we take these therapies forward.
That's terrific. Thank you, Dr. Barker. And if our technology allowed a two-way exchange at this point, you would hear a chorus of applause from coast to coast and beyond that would gratify you, and it certainly gratifies us to have had you leading this terribly important talk. Thank you so much. I have a few announcements. Great if you stay on the line for a second, Dr. Barker. We'd probably like to debrief you anyway for a minute afterwards. But for those of you on this call, the usual routine, for those of you who've done many of these, do please complete the online survey that should be on your screen at this point. You know that we do take this very seriously, analyze it each time, and it does help us improve the webinars and also make subject selection as we go forward. So please do fill in the online survey. I will repeat our gratitude to AbbVie Incorporated and Lundbeck LLC, the two major pharmaceutical companies that have made this series of talks possible in this season, where they've done this with very much with generosity, but also with the understanding that we determine what the content is, which makes the whole thing both kosher and also extremely interesting and important to everybody who participates. So thank you, those two great companies. I should tell you there's an archive of the event which was made available starting next Tuesday, which is April 12. You can find it at our website, the Parkinson Disease Foundation. It's www.pdf.org. We'll send you an email with the link when it's available so you can listen to the talk again, or of course let your friends or others in your family also share this. Save the date. We have the next in the series is coming up on Tuesday. It's always a Tuesday, June 14. That's 1/4/2016. It's going to be, as usual, from 1 p.m. to 2 p.m. Eastern Daylight Time in the United States. The topic will be apathy or depression. Which one is it? And it's going to be led by Dr. Dawn Bowers, who is a professor of neuropsychology at the University of Florida and director of the Cognitive Neuroscience Laboratory at the McKnight Brain Institute. So please join us for that on Tuesday, Tuesday, June 14. So with that final thought, just to repeat our thanks in behalf of all of the people listening today and those who will tune in the next month. Dr. Barker, this was really an outstanding presentation. I'm afraid we're going to be have you back in one form or another, so maybe you can shut your phone off or have it reject our calls, because definitely you're going to be in great demand, even greater demand than you already are, and we're very grateful for this. And I, for one, am going to be going to the whole talk again because I think I missed too much on the way, and it's that kind of talk. Beautifully done, very important, and to all our participants in this talk today, thank you for joining us, and come back next time. Goodbye. Enjoy your spring when it eventually comes, and we'll see you in June, or hear you in June. Bye-bye.